Once this video is completed, please review and answer the following goals. DNA stands for deoxyribonucleic acid and is found in all forms of cellular life. Recall that viruses do not contain cells, and even though some may have DNA, not all have DNA and may have RNA instead. Sometimes DNA is found in cells in the form of chromosomes, structures of DNA organized with protein. DNA has specific segments located within it called genes. Genes provide the framework of the development and function of an organism. A genome, however, is the entirety of genetic information, that is, DNA, found in an organism. To the right is a representation of the genome of the bacterium E. coli. Fruit, such as strawberries, have DNA as well. Did you know that different numbers of chromosomes can be found in different types of strawberries? Native strawberries usually have two sets of chromosomes and are thus termed diploid. However, cultivated strawberries, the ones that can be found in a typical American supermarket, have 8 sets of chromosomes and are termed octoploid. There are several steps that you would take to extract DNA from a piece of fruit such as a strawberry. In lab, you would get a strawberry and put it in a plastic bag similar to a Ziploc bag. Once it's in the Ziploc bag, you would seal it up and then squish the bag until you generate a type of fruit puree. Once that's done, you would measure out 10 mils of a lysis solution. A lysis solution usually contains some kind of soap with water and maybe some salt. Once the lysis solution is measured out, you would open the bag and put the lysis solution into the bag and squish together the strawberry and the lysis solution. You would try to do this in a gentle way, trying not to create too many bubbles. A filter apparatus should be set up with a clean test tube and a clean funnel with cheesecloth inside. The cheesecloth inside of the funnel will allow for a filtering device. Once the lysis strawberry solution has been thoroughly mixed, it is then poured into the funnel with the cheesecloth and is allowed to drip into the test tube. There should be at least 15 drops that fall into the test tube. After 15 drops have been added to the test tube, you would then add DNA precipitation solution to the test tube until the test tube is about half full. The precipitation solution usually consists of some kind of alcohol that has been chilled prior to an experiment for DNA extraction. Once the alcohol or precipitation solution has been added, you would observe for 3 minutes. This is to allow separation within the solution. Once 3 minutes has elapsed, at the top of the solution, you might see something that looks thick and viscous compared to the rest of the solution. This is what we call the DNA. The DNA is usually thick, and we usually remove it with some kind of wood, stick, or toothpick. You will also see tiny bubbles begin to form within the DNA. Normally in lab, we would repeat with another living organism. So this can be a piece of fruit, this can be chickpeas, this can be bacteria, this can be anything that is composed of cells. This is what you would expect from a typical DNA extraction. The right image shows a DNA extraction from germinating peas. The left image shows DNA extraction from strawberries. Notice how they both have a color solution on the bottom, which is what we refer to as the lysate. At the top, where it's separated from everything else, or starting to separate from everything else, where it looks like a cloud or thin wispies, those are what we refer to as the DNA, and it's also associated with protein. Once you've gone through this whole PowerPoint presentation, Please make sure to review the goals thoroughly, make sure that you can answer them, and go through your manual and answer both the pre-lab and post-lab questions. As usual, please email your instructor if you have any questions.